Hello everyone, we'll start with new chapter, the last one in fact. It is the economic importance of bacteria and fungi. Firstly, we'll cover the topic of bacteria. So, economic importance of bacteria. Before beginning with that, we must know what bacteria is and what does it look like. So, bacteria is the most primitive. Primitive means it was the first one to be formed. It was formed quite earlier. The most ancient one is bacteria. And it is unicellular. Unicellular means it is just one cell, single cell organism. And it is not covered by its DNA, its nuclear material. The DNA of bacteria is not covered by a nuclear membrane. Basically, when we talk about animal cell and plant cell, we draw nucleus inside it, and inside that is present the DNA or the genetic material. So basically, this membrane is present in most of the organisms, but it is missing in case of bacteria. So it is not, its DNA, its genetic material is not wrapped by a nuclear membrane. It is not wrapped by any membrane in fact. So such organisms are known as prokaryotics. So bacteria is a prokaryote, which means its DNA is not covered by any membrane. Alright. Talking about where is bacteria present, bacteria is present almost everywhere. Wherever, if you talk about air, water, soil, and food, inside living organisms, on living organisms, they are practically present everywhere. In fact, we don't know how much number of bacteria are just present right now on your body, on your skin, on your hands, okay? But some of the bacteria which are present inside our body, they are quite useful. There is one bacteria, the name of it is E. coli, Escherichia coli. This bacteria is present in our intestine and it helps to synthesize vitamin B. So this bacteria is quite useful for us, quite important for us, right? Now talking about size, the bacteria is the smallest living organism. Its size ranges from 2 micrometer in case of length and 0.5 micrometer in case of breadth. This is quite a small size. Talking about shape. So shape, if the shape of bacteria is round, we call it as cocci. If its shape is rod-like, we call it bacilli. If it is spring-like, we call it spirilla. And if it is comma-shaped, then we call it as vibrio. These are the shapes of the bacteria, and mostly bacteria are present in groups. So if two spherical bacteria are present, we call them as diplococci. If they are present in a chain, if the spherical bacteria they are present in chain, we call them as streptococci. And if they are present in cluster, a group, then we call them as staphylococci. Staphylococci, right? Next is that how does the general structure of bacteria, how does a bacteria look like? We'll draw a general structure. So bacteria is having a cell wall which is made up of peptidoglycan. The cell wall of plants, they are made up of cellulose, but the cell wall of a bacteria is made up of peptidoglycan. Inside the cell wall is present the cell membrane. This is the cell membrane. Sometimes the bacteria is further covered by a hard structure outside its cell wall, and that is known as capsule. For further protection, even its cell wall is covered by a material which is known as capsule. Okay, and inside this bacteria is present the genetic material, the DNA, right? All this is cytoplasm. It is having all the organelles like vacuoles, etc. Okay, and mostly this bacteria, they cannot move, they do not show locomotion, but they are carried away by wind and water. But some of the bacteria, they have structure known as flagella. One is known as flagellum, plural is flagella, right? So some of the bacteria, they move with the help of these flagella, okay? But most of the bacteria, they cannot move, they are just carried along with wind and water and etc. Okay, so what does this bacteria eat? So it is not having any chlorophyll or anything like that. So bacteria basically feeds on other organisms. Such organisms are known as heterotrophs, which depend on others for food. So bacteria which basically feed on dead and decaying material, they are known as saprophytic. And the one which lives on living organism and takes its food from it, they are known as parasitic, right? Talking about respiration, bacteria can respirate in the presence of oxygen as well as in the absence of oxygen. If bacteria is respirating in presence of oxygen, we call it as aerobic respiration. And if it doesn't need oxygen, it can breathe without oxygen, it can respirate without oxygen, then that type of respiration is known as anaerobic respiration. Now, how does bacteria reproduce? Bacteria reproduce, just what happens is that one bacteria, its DNA, firstly, it duplicates. This is one bacteria, this is DNA material. The first step is duplication of DNA. DNA becomes double. Next step is that this DNA, which is duplicated now, it will start separating like this. And finally, this furrow, this is bacteria, its DNA started duplicating itself. After the duplication, the DNA started moving on the extreme ends and there is a formation of furrow. Ultimately, this furrow, it breaks down and bacteria divides into two. So simply, one is being divided into two. This is known as binary fission. Binary means two. And fission means splitting, breakdown. So one is breaking down into two. This is the major way of bacteria to reproduce. Okay. Next is spore formation. So if bacteria cannot survive some of the situations, in that case, bacteria develops a hard layer around itself. And it does not show any activities. It does not feed, it does not excrete out the waste materials, it starts collecting the waste material even within itself. So such conditions are like there is extreme dryness outside, or say there is a very high temperature, or if bacteria, feels that there are quite harmful or poisonous chemicals outside in its environment, if it feels like, if it senses the presence of harmful poisonous material, or if it senses that the temperature outside is too high, in that case bacteria wraps itself up in a layer, in a very hard structure, and bacteria takes the shape of a spore. This is a spore, and bacteria will not let itself come out until and unless all these situations go away, pass away. These situations are known as unfavorable conditions because it cannot tolerate very dry weather, it cannot tolerate very high temperature, and it cannot tolerate any of the harmful substances. So these are unfavorable conditions for the bacteria. It cannot grow in these conditions. In that case, bacteria takes the shape of a spore. This is a round body which is very hard, that it just cannot be broken down. Even if we heat a substance which is having the spores of bacteria, no matter how many high temperature you give it to, this layer will not break at all. Okay, so bacteria saves itself from the harmful conditions, or say unfavorable conditions, by turning itself into a spore. Alright, now talking about the uses of bacteria. Bacteria plays a major role in medicine. So a lot of antibiotics, serums, and vaccines are prepared from the bacteria. So talking about the discovery of antibiotic, the very first antibiotic to be discovered was penicillin, discovered by Alexander Fleming. Alexander Fleming was growing some bacteria in laboratory. He observed after a few days that the growth of bacteria has been paused, or the bacteria are being killed because there is growth of some else material, which was a mold, a fungi. A mold started growing where the bacteria was being cultured, being grown, and this mold secreted a material which was inhibiting the growth of this bacteria. It was not, this secretion by the mold was not letting the bacteria to grow, and he named this secretion as penicillin. So he said that there are some materials which are secreted by someone else, but these materials do not let the harmful bacteria to grow. So he said, he named them as antibiotics, and penicillin was the first one. But penicillin was not discovered from the bacteria, but it was a mold, it was a fungi. But after that, a lot of similar antibiotics were made from bacteria like streptomycin. There is one table given in your book that you have to learn. In fact, you have to learn the name of the antibiotic, from the source from where that antibiotic is produced, and which disease it is used to cure. For example, penicillin is used to cure tetanus. Okay. Now, what makes a good antibiotic? Antibiotic is good if it does not kill only one type of bacteria but a number of different bacteria. Then we call it as a good antibiotic. Secondly, if it does not produce any side effects, then that antibiotic is a good one. And antibiotic is good only if it kills only the harmful bacteria and not all the bacteria which can act as useful as well. So number of bacteria are present in our body, some of them are useful, some of them are harmful. Antibiotic is good only when it will kill the harmful bacteria and not all the useful ones. Okay. Antibiotics are also used for preserving meat and fish, for also controlling the plant pathogens, and etc. Next are the serums. What is serum basically? You all know what is blood. Blood gets clotted, blood forms clots. There is one material present inside the blood which is known as fibrinogen. This is the material that forms clot inside the blood. But if we will remove this material from the blood, then what is left is known as serum. Blood minus fibrinogen. Fibrinogen is the one which forms the clot. If we will minus fibrinogen from it, then the blood will not form any clot. So it will stay as a liquid, and that liquid is known as serum. So what happens is that a harmful material is selected. For example, poison of a snake is collected. A very little quantity of that poison is injected in the body of a horse. The blood of horse will try to kill this toxin. Toxin is a harmful substance. It will try to kill this toxin, and its blood will produce antibodies. When our body cures us naturally, it forms antibodies. The WBCs of the blood form antibodies. Okay, we injected a very small amount of toxin in the body of horse. The horse blood, it reacted and formed antibodies. Now we will collect the blood from this horse, remove fibrinogen from it, and we will store its serum. Now this serum is having antibodies against this toxin that we injected inside the horse. Okay, so this blood now is stored in the laboratories or in the hospitals, and whenever there is a need, in that case this serum is directly injected in the person who is infected. For example, a person got stung by a snake. Okay, a snake it has bitten a person. Now person needs a immediate treatment. For that, this person is taken to the hospital. Okay, then doctor will ask that person, what kind of snake was it, how does it look like, and etc. All the questions he'll ask to the patient, so that he can pick up the serum from the hospital and immediately give an injection to that person, right? How the serum was prepared earlier, was that all the types of snakes were chosen. Snakes were then, the poison from the snake was collected. Then that poison was injected in the horse body. Then the blood of the horse was collected. Now if, let us say, we take an example of any snake, let us say anaconda. Let us just take an example. Let us say anaconda. We gave the poison of it to horse. Now horse produced the antibodies against anaconda only, right? So the doctor will ask the person what type of snake it was, so that he can pick that particular serum with that particular antibodies that have been collected, and then person will be relieved immediately, because antibodies which are already developed, when it enters the body of the person, they will immediately detoxify the poisonous substance. Toxin is the harmful and antitoxin is the one which works against it. So the person will immediately get relief, because the readily prepared, the artificially prepared, or say naturally prepared antibodies are given to the person who is attacked by a snake, and then that antitoxins will kill the harmful poisonous substance that has entered inside the body of the person, and person will be saved. Similarly, the vaccines are prepared. What is vaccine is that, remember vaccine, we are now even inside, in case of corona, the vaccines are being prepared. So what will happen in case of vaccination? What happens in case of vaccination is that whatever disease has been caused to you, if you want to get cured from that disease, for example, you are vaccinated against, say, measles, typhoid. Let us take the example of typhoid. So if you are vaccinated for typhoid, it means the germs of typhoid has been injected inside your body. If you are taking a vaccine of polio, it means the germs of polio are injected inside your body. But these germs are sometimes dead, or they are very weak, so that the weak germs are injected inside your body, so that they do not cause you polio, but they will let the blood itself to form antibodies against this polio, so that in future, if an active germ of polio enters in your body will already be having antibodies against it, and it will immediately kill the germ of polio. Okay? When your body is vaccinated, your body is being injected by weak or harmful, not harmful, harmless, by very weak or dead or very little amount of bacteria. They are injected inside your body, so that your body naturally produces antibodies against them, so that in future, if that same germ is attacking your body, then your body will naturally be saved. Alright, so vaccines are also prepared by the bacteria. And bacteria is also playing an important role in agriculture. Sometimes the plants, they cannot take the nitrogen directly from the atmosphere. And if in the soil there is less amount of nitrogen, then there are some bacteria present in the soil. They are known as nitrogen fixators. They fix this atmospheric nitrogen into nitrates, so that the plants can use these nitrates. Okay, such bacteria, they are known as nitrogen fixing bacteria, and this process of converting nitrogen into nitrate, this is known as nitrification. Some of the bacteria are present in the soil which convert this nitrate back into nitrogen, and this process is known as denitrification. Okay, So there are some bacteria which converts nitrogen first into ammonia, then into nitrite, and finally into nitrate. And there are some bacteria which converts nitrogen directly into nitrate. Okay, and then there are some bacteria which convert nitrates back into nitrogen. Okay, so this cycle continues, and this cycle is known as nitrogen cycle. Okay, then bacteria is also used for preparing wines and etc., and giving flavors also to tea and coffee. Now bacteria, if they are useful, they are harmful as well. They, if they are present on any of the food, then they cause food poisoning. If bacteria continuously grow on a food and we eat that food, then we suffer by food poisoning. This quite high risk of food poisoning is known as botulism. Botulism, food poisoning. Okay, so if we want to save yourself from food poisoning, you just make sure that the food has been properly preserved. So there are some steps of food preservation. In order to kill the bacteria, we heat a particular thing, or we boil it, or we heat it at a high temperature so that bacteria doesn't grow on it. Next method of food preservation is that we add salt to it. All the pickles, we add salt to pickles so that they do not get destroyed by bacteria. Then dehydration, we dry the thing. For example, milk powder, milk gets ruined after a few days, but dry milk, it can continue till a long period, because some of the bacteria, they cannot grow without water. So if you are dehydrating a thing, removing water from it, then that thing can go up to a longer period. Then we kill the bacteria by UV rays. This is also one of the method. Then there is pasteurization. What is done in pasteurization is, first the milk is heated at 60 degrees Celsius. Then after heating it at 60 degrees Celsius, it is immediately put in freezer for chilling it. This process is pasteurization. This also kills a lot of bacteria. Then refrigeration. We all know, in order to preserve food, we keep it in refrigerator. It can go for 2-3 days long, right? Yeah, that's all about bacteria. Some of the bacteria are present in plants as well, and they cause diseases in plants, and some of the bacteria cause diseases in humans, like cholera, tuberculosis. They all are caused by bacteria. And then bacteria, some of the countries, it has been said that some of the countries are preparing bacteria, like this corona. This also was, firstly the news came that this is acting as a bioweapon. China prepared this bacteria, this virus, so that it can attack on other countries, but it got infected itself as well. So countries, they prepare some of the bacteria such that such bacteria cannot grow in their country, but in their, what we call it as, one which they are not having a good relation with. Like China is not having a good relation with India, so China will prepare such a bacteria that it can only affect the people of India. Not only India, but worldwide effects. We are seeing now this in case of corona as well. This is going worldwide, this pandemic. So such bacteria which are prepared in as germ bombs, so that the disease can be spread in the other country, they are known as bioweapons. That we are not fighting with, we are not going at war with other countries via guns and all, but we are going with biology. Such weapons are known as bioweapons. That's all. We'll continue in the next topic about fungi.